So I work up at Mount Sinai in New York, not Mount Sinai in Miami, which has been a common source of confusion. And so the four of us actually just started a lab. We've been working there for about 4 years, but we finally have carved out a space where we work exclusively on personalized cancer vaccines. And I'll talk a lot more about what that means. But the basic idea is that these are, so personalized medicine in general, and specifically what we're doing, it implies that there's going to be a strong algorithmic component. So typically what you do with medicine is you have a thing that you think works for some disease, and so you run a trial, and then you see if, among the 1,000 people on average, was there a difference between two groups. And that's sort of normal medicine, average medicine. As soon as you are quantifying individuals, there's no possibility of running a trial, because the idea is that something's going to be different about that person from everyone else. So what you instead need is you need a prediction for whatever data you've quantified from the individual. So it's kind of a weird new regime of medicine. And it's been interesting to see so the FDA is actually pretty accommodating and inviting of this new modality, while also being really cautious about really deploying it en masse. So I think they're very welcoming of weird clinical trials that heavily personalize therapy. And then we'll see in a couple years how they feel about actually making this available to the general public outside of clinical trials. So this is kind of a new way to do medicine. And so we're all computational. Everyone has like a degree in computer science or math or something like that. And what we do is we develop software and models for making vaccines against people's cancers one patient at a time. And that software is all up on GitHub. So it's a very computery culture. And doctors are really confused. I don't think any of them know what GitHub is, but whether they know it or not, there are 3 clinical trials that we're running at Mount Sinai that run on our software. And so there's kind of a weird merging of the worlds. An oncologist may see the patient up until the point where we get the data from their tumor sample, at which point a lot of Python scripts come to life. And doctors slowly have to learn about what those are, and that that's really a central piece of treatment. So really quick as I can make, background in cancer immunotherapy, which is sort of the bigger category that the stuff we work on falls into. So starting with just what is cancer. And so I kind of think of cancer as like a rebellion in the body. So the body is a very rigid police state. Every cell has a very limited set of activities it has to engage in relative to what it could do. And particularly the division of cells is really tightly controlled. So most cells, when you get older, only a few kinds of cells are allowed to replenish themselves at a very limited rate. And most cells kind of sit quiescently and just do their thing, whatever that thing is. They make some insulin, they make some melanin, they're just doing their thing. And if they stop doing their thing, if they start misbehaving, there is an elaborate infrastructure of an immune police state that kills them. So one of the main roles of the immune system, we think of it mostly as being about bacteria and viruses and stuff. One of the central roles of the immune system is killing misbehaving cells to keep the order of the body, because really you're not quite one organism. You're like billions of organisms that are being forced to cooperate. And so what happens, either due to environmental exposure or just over time by chance, is that all the cells in your body are mutating just a little bit. Like each cell division is a chance to mutate a little bit. And most of those mutations are totally innocuous. So you take a very healthy organ and you sequence it, and you find that every cell has a little difference, and it didn't really matter that it had that difference. But just by chance, one cell in your body somewhere over time is going to accumulate mutations that aren't so innocuous. And there's a moment where those culminate in just a total neglect of the protocols. The cell doesn't check with its neighboring cells to see if there's space for it to divide. The cell doesn't check to see, are there nutrients for me to divide? It doesn't really care about producing the thing it was supposed to. It just starts making more of itself. And if it does that and acquires some other kind of properties of moving around, and it really sort of becomes a separate organism. And that is a cancer. And so there's a lot of subtlety in defining that, because there are lots of things that aren't cancers that are a little bit different from you. But when it becomes a problem, when it really endangers your health, then we look at it and go like, okay, that used to be you, but now it's just its own thing. And we want to get rid of it because it's not cooperating with the rest of your body anymore. And so a big discovery that's been like slow building, I mean, really the first work in this is in the 1800s, and it was totally ignored forever. And then it had a resurgence in the past like 20 years, is just the role of the immune system in controlling this stuff. Because you're constantly having little deviations throughout your body. And sorry if that's like spooky language. Maybe I'll say it a little more indirectly. But like, there are always little pockets of cells that are kind of going weird. They're doing their own thing, and the immune system sweeps in and kills them. And that's the most typical thing is this column A is there's some weird cells and a bunch of immune cells show up and they kill them and you go back to being normal. A much rarer event that happens over our lifetimes is sometimes there's pockets of weirdness that hit equilibrium of the immune system. It doesn't quite kill all of them. They sustain at a low level. And then there's a small chance that when you're typically much older, that little pocket of weirdness finally gets to escape from the immune system. And so there's a really famous paper called The Hallmarks of Cancer, I think it came out in the 80s. And then this is a chart from the updated Hallmarks of Cancer, which has these two new categories, which are immune escape and immune suppression. And that's starting to become something that's just, if you look at any cancer that's detected, that if someone has a cancer and you look inside of it, it's very common that to become that cancer, to escape the immune system's police state, it needed to both figure out how to hide, how to not present itself to the immune system like normal cells, and if caught, to suppress the immune system, to tell the immune system to go away. And so these are starting to become things that are thought of as just definitional, like to be a cancer, it needs to be able to like dodge the immune system in order to have grown to a certain size. And so that line of thinking leads to a lot of new therapies. So instead of trying to kill cancer cells directly, instead you can do things that treat the immune system to get the immune system to be more aggressive or to be more selective. So if the problem in a patient was that their immune system was being suppressed, you can try to block that suppression to make the immune system do its thing more aggressively. If the problem was that the cancer somehow evaded detection, then you can try to reorient the immune system and say, okay, look over here. This is the thing you should be killing. And so I'm going to go to a chart that's a survival curve, and I don't want to oversell it because this isn't really a done deal. This is really new research that for a lot of cancers doesn't work. But there are a few cancers for which this is pretty dramatic. And so this is, these are like the bread and butter of cancer research, and they're also terribly depressing because they're plots where the y-axis is the number of living patients. And so for metastatic cancers, the standard treatment is a curve that goes to 0, and the thing that you're looking for is a slower progression to 0. So chemotherapy and radiation therapy, if you apply them to non-localized cancers, cancers that have really escaped where they started, it's very typical that you get a curve that just goes down, down, down forever, and a successful clinical trial is a gap between these two curves where just the better drug, the patients lived a little bit longer. And so a thing that's starting to appear in these curves as people experiment more with immunotherapies are asymptotes, plateaus where a large fraction of the patients don't die. And that's actually pretty new for metastatic solid cancers. There's a few exceptions where there are good treatments, but for like metastatic melanoma, it's really very strange compared to the research that was done in the 70s, 80s, 90s. And there was a lot of research. There was like decades of trying different targeted agents and chemotherapies, and they all didn't have this property, which is that you actually get patients that are just going to live roughly as long as they would have lived otherwise, which doesn't mean that they're cured. Often what it means is that they've kind of hit that equilibrium where if the cancer tries to spread, the immune system gets the spread. So they might still have some pockets somewhere, but it's not deadly to them anymore. And so that's like created a really big change in cancer medicine. This like realization that if you treat the immune system, then you can get a very large fraction of patients to live, even if they started out with cancer all over their body. because the immune system is going to go all over their body and find all those cells and either kill them or lock them into little pockets they can't get out of. So this is like a success case. This is a drug that makes the immune system more aggressive without really targeting anything specifically. Okay, so the stuff that we work on is a sort of complementary side of that, which is like, what if the problem is just the immune system doesn't know what the cancer looks like? And so I'm going to unpack some of the terminology. So a therapeutic cancer vaccine, so vaccine I think conjures like you get a jab for the flu or something. So a therapeutic vaccine is one that's actually trying to clear an established disease. You're not trying to prevent something, you're trying to just get the immune system to look at this example of like, this is something bad, and then kind of pattern match that onto all the cells of your body to see like, does that look bad? Does that look bad? Because the immune system itself is like a learning algorithm. And vaccines are like training the learning algorithm of your immune system. You give them examples paired with some label that says this is bad, and then the immune system learns it and goes around and kills everything that matches it. And then a therapeutic cancer vaccine is a vaccine whose aim is to get the immune system to sort of rise up against the cancer. Okay, so what's in one of these? This is like a little bit of a technical slide, but the two major components are there's an antigen, the thing the immune system is supposed to recognize, and then often there's an adjuvant, which is some intrinsic labeling. It's a thing that tells the immune system this is bad. And it could be anything that looks weird. You could add like killed bacteria, they look weird. You could add double-stranded RNA, that doesn't happen in the body. So anything that looks really weird, you take some examples of stuff that's in the cancer and you mix it with stuff that the immune system knows for sure is bad, and that could potentially cause that connection to be made within the immune system's memory. So adding in one more term, so personalized therapeutic cancer vaccines are cancer vaccines which get made from scratch for each patient. So rather than trying to make a vaccine for melanoma, you just take the tumor and look at it somehow. You profile it, and then you make a vaccine for that patient, which is laborious and expensive, but potentially more effective. So how do we choose what goes in? Right now, the most common thing is genomic sequencing. So kind of like 23andMe, but a lot more data. And we do that sequencing both for the patient and for the tumor because their genomes are different. And we're looking for what is different between them. And once we found that set of differences, then we're going to filter the mutations, and the filtering is where the machine learning comes in. So before I get into the details of how we do it, I'm just going to show a little bit of the history of experiments people have done. So one mouse experiment, then one human trial. And this stuff's really new. This is a paper from 2016. And so this is really densely packed, but the two things I want you to look at are this plot here where they're counting the number of detectable bits of cancer in the mouse's lung. So the lungs look like this, and I think if you just look down here, you might see ones that look kind of normal and ones that look horrifying. And so these are mice that have had cancer implanted into them, and then it spreads to their lung. So this is like a metastatic model. And if you don't treat it, then the lungs get covered in cancer. If you treat these mice with one version of the vaccine, this like Epitope thing, it's 5 mutations that they're being vaccinated against, it sometimes works to reduce the number of these tumor nodules in the lung. And there's a different set of 5 mutations, this Epitope 2, that always works. So this is kind of evidence that it actually matters quite a bit which mutations you choose. And these Epitope things, these groups of 5 mutations, were already kind of chosen for some heuristic of ones that they thought were good. So just being a mutation that's in many of the cancer cells that has like a lot of expression, a lot of it's being made, is not enough. You need to predict something about how the immune system is going to see the mutations in order to get the one where it's always working rather than sometimes sort of working. Okay, so human trials, there's a whole bunch of human trials going on now, but they're too recent to really have too much data. So there are 2 that have published so far that I know of that actually have some hint of it's working. And so this is one of them. This is at Dana-Farber. It's a trial run by Kathy Wu, who's like one of the head honchos of this field. And so they published on 6 patients from their trial. And so in those patients, what you see is it's weak evidence. Like, we don't know for sure whether this is working, but you get kind of a hint. So you had stage 3 melanoma patients, and after they get vaccinated, their cancer did not go away, but it stopped spreading. So that's kind of enticing. Like, they didn't advance to stage 4, which is what people normally would do. And then there were 2 stage 4 patients for whom the vaccine did not immediately work. They actually started progressing, but then they were treated with a second immunotherapy that made the immune response stronger, and then they had complete remissions. So they had complete responses. So that's like an even more tantalizing hint of like, this sometimes is enough to sort of stop the spread, and then maybe you could amp up the response. You can make it more aggressive, and that'll actually be potentially curative. So this is a very exciting but tiny data set of patients. All right, so now I'm getting into the actual machine learning. So where does the machine learning fit in? So this is kind of a typical funnel of potential targets. There's a lot of mutations, but if you filter them down to just the ones that seem to change proteins, then you'll get maybe 50 or 100 or 200, sometimes 1,000 for a melanoma. And you can filter a little bit more, but you still end up with more mutations than you want to put in a vaccine than is practical to. And so that is the role of machine learning. So this last step where you're taking some mutations and then you're ranking them by will the immune system see them is the sort of final filter of what goes into a vaccine. And so a little more biology is just to explain what the models are. This is a ridiculous diagram, but in this diagram, a protein in your cell. So the proteins are just the machines that are going on inside of a cell. Everything in a cell is essentially a protein of some sort that's active. They're always being chopped up and recycled. So in this diagram, that's the chicken. And when they get chopped up, you get little bits of them, peptides, little short fragments of proteins that get put onto this presentation platform called an MHC. So it's just it's like a little hot dog bun that's pasted all over the surface of every cell that in it has a little piece of a protein. And the reason that every cell has to do this is because they're being surveilled by the immune system. Specifically, T cells are looking for, do you have some hot dog buns with the right thing in it? And the right thing from the T cell's point of view is going to cause the cell to get killed. So every T cell's got its own notion of what's the bad thing I'm looking for, and when they find that thing, they kill the cell. So you might ask, why do the cells even bother? There's a different cell type called natural killer cells that go around looking for, do you not have enough of the hot dog buns? If you have too few, then the cell gets killed. So they're sort of trapped in this if you don't show anything, then you get killed. If you show the wrong thing, you get killed. And this is why I think of it as a police state. And so one of the central prediction problems is what is going to fit inside of these MHC molecules. So this is a structure of an MHC. It's 300 something amino acids. And there's a little green bit on top that's it's a piece of some other protein that'll wedge in pretty firmly in some cases and usually just flops right out. So predicting whether or not some piece of a protein is going to fit is the problem. There's a lot of other problems, but this is maybe the most important one as far as determining whether or not the immune system will kill cancer cells. So this is the immune epitope database. It's very lucky that a bunch of immunologists started doing this, because I don't know if they had specific applications in mind, but I think in the 80s they started pooling their data, and then eventually in the 90s they put up a web front end, and there's this database of just a lot of immunological data that's really useful for building machine learning models. And so I'll show you just examples of what some of the data looks like. So that first column are it's a label on which kind of MHC. There's thousands of different MHCs. It's not one problem. It's actually thousands of related problems, and they each have a name. And then this epitope description thing is the sequence of the piece of the protein. So what you're trying to do is you get a sequence out of an alphabet of 20 letters, and the length of the sequence is between 8 to 15 amino acids, and you're trying to predict this number. So like it has a lot of bio background and sounds complicated, but it boils down to like a pretty simple problem. You need to have a model that takes a sequence as an input and spits out a number. So that's one of the sort of core data sets and the core problems. So a slightly different kind of data set, and this is something we're still trying to figure out how to really use, is there's also data on what if T cells recognized. And it's sort of similar in style. So there's an MHC column that says the name of the MHC, and there's a peptide column that tells you the sequence of that little piece. But here the response that you're trying to predict is, did T cells trying to kill something showing this peptide MHC complex? So these are our data sets. And so one thing that's kind of surprising is that for the MHC binding problem, linear models are not bad. They're like the classical mainstay of immunologists trying to predict this stuff. Been used since like the 80s. And so a linear model doesn't perform too badly. Neural networks do better. So there's this tool, NetMHCPan, they're all very poorly named. So this takes a 9-letter input, and then it has 1 number as an output with a bunch of neural network magic in the middle. And now that deep learning has taken off, there's like a proliferation of papers that are different deep learning models. So convolutional models, recurrent models, all sorts of goofy models that are trying to predict these problems with like larger architectures. But if you look at the toolkits, like what you're using to solve these problems, the immunology really disappears quite quickly, because what we do and what a lot of the groups are starting to do is they're using the same tool set as any other kind of data science problem in Python. So we use IPython and we use pandas to load all the data, and we will visualize the data with Seaborn. And if we want to do sort of basic machine learning stuff like cross-validation, that happens through scikit-learn. That's also our baseline of linear models we train through scikit-learn. And then once we start crafting deeper neural networky models, we switch to Keras, or I'm starting to use PyTorch more. But none of this is really specific to biology or immunology or oncology. These are just the like generic Python data science stack. And so as a last slide, because I think it's kind of a more businessy audience, I wanted to point out this is like a huge startup bubble. There have been, I think, I can't even count them anymore, but maybe 30 companies at this point, which are seemingly all getting funded pretty well. So I highlighted some of the better known ones, but this is really a rapidly evolving startup scene where often the differentiation between the companies ends up being the sort of machine learning savvy that they often don't have. So they all kind of started pitching like deep learning expertise, and I've consulted for 2 of them, and I'm aware that they do not have deep learning expertise. But they know that it's a very important part of the pipeline. And so it's forcing them to go out into communities like this and sort of research communities like NIPS and ICML are the 2 big machine learning conferences. So you're getting an increasing showing of companies that do personalized cancer therapies that previously would have looked quite out of place, but now are like merging fields. And I think that's it. We do questions?
Yeah, depends on your.
Yeah.
Thanks so much for a great panel.
I have a question for you. It's more a bit more philosophical than not trying to keep it really short. You use a lot of open emerging frameworks in order to do sort of develop vaccines and make it more personal. And efficacy rate is usually very, very precise. For instance, with HPV vaccines, like 1 in a million, right? That you get something that you have an adverse event. But when you look at commercial, customer conversion, churn rate conversion, those retail models, those get it right maybe 4 out of 5 times. 1 in 5 they don't get it right. So how do you develop that same kind of precision in more commercial, retail, those sorts of models that matches this tremendous sophistication that you have in these sorts of clinical therapies?
I mean, I think one answer is that, particularly for personalized medicine, you don't really know very much. There's not Often efficacy is scattershot. That's why you have a lot of clinical trials that are indistinguishable from the control arm. We don't know what to predict. I think it's wrong to compare with prophylactic vaccines, which is one of the most conservative, carefully developed fields in medicine, because you're treating healthy people. So I think that preventative vaccines, they advance glacially. I mean, there's 4 adjuvants that have been approved by the FDA ever, I think, and it's the ones that people have used, I think, since the 50s, and maybe one new one in the 80s. And so it's very, very slow and careful. And in cancer, on the other hand, I think people are often just guessing, because there's not really time to get it right, and there's patients with high need. So if you look at clinical trials, they're not careful. They're sort of like, oh, this looked really good, and we tried it on one person, it seemed to work, so let's make a trial out of it. So I think that there's an inversion between treating a healthy population, where you need to almost never do harm, versus treating people who are almost certainly going to die, in which case there's a much more of a risk appetite by those patients. And I think within oncology, that's a gradient, because if you're treating patients who have localized disease for whom surgery is probably curative, then you want to behave more like preventative vaccines. And if you're treating patients without good treatment options, like glioblastoma right now doesn't have good treatment options, then people try pretty random shit. And they wouldn't phrase it that way. They would tell it, we have 20 years of preclinical models that have justified the use of whatever. But when you really dig into it, they're kind of trying random shit. And it hasn't worked yet, and so they're going to try more random shit.
Thanks, man. Great talk. Just a quick question. Anytime you're dealing with omics, you're dealing with large amounts of data, especially with genomics, and you start to run into the possibility of spurious correlations.
Mm-hmm.
What are you doing with your data to reduce that risk? Are you using dimensionality reduction, or are you doing a von Ferroni correction to your data to try to reduce the risk of spurious correlations?
So the kind of things that we do, we're not really doing sort of GWAS-y type studies. So a common thing in biology is you look at all the mutations in a big population and you try to figure out which of these is responsible for people being blind or something. I don't know, just some random phenotype. So we typically don't do anything like that, because we're working one patient at a time, and we're not really, so within the sequencing data per patient, we're relying on models that have been trained on actually pretty simple data sets. And that's that might actually be the answer, is that you don't want to do something that looks at the whole genome of a patient and then apply a model that was trained on a lot of whole genome data from other patients, because then I think you will have crazy false positives. So what we instead do is we use these fairly restricted data sets, 100,000 samples whose, it's short sequences mapped to floats with where neural networks with 40 hidden layers might be too much capacity, so we scale it down. So I think we try to use restrictive models with a lot of testing of each assumption and stack those up, rather than doing something where we take all the genomics data in the world, make some big model, and then apply it to a patient, which would be much scarier. And I think that's a problem maybe in general, in personalized medicine. If you wanted to just sort of look at all the people with different diseases and all the drugs they've taken and then make some model out of that, then I think you'd run into a lot of weird false positives that would be hard to ferret out.
Thank you so much. My question kind of focuses on some of the outcomes. Have you guys looked at some of the patients who have problems with responding to this, to see if there are specific markers that kind of herald, especially the cytokine storm and some of the other immediate reactions to the treatment?
So the problem with cancer vaccines so far is more that they don't work rather than they have strong reactions. So it's not like cellular therapies that induce cytokine storm, or checkpoint blockade can induce colitis or systemic inflammation. Cancer vaccines mostly don't work. So this is kind of a new sort of cancer vaccine for which we have a little bit of data where even that little bit, it still looks like it's like skewing too far towards the not killing enough rather than like getting out of control. So yeah, the thing we're trying to figure out is like, how do you get it to work better, which I think is going to then get into more safety problems, because that's probably going to imply some like, one of our trials that we're starting this month is a combination with a checkpoint blockade agent. So it's going to be vaccine plus all the T cells go crazy at the same time. And I think there we might have bigger safety problems. And a lot of the startups are doing that too. And I think that like in a year, people will start publishing a lot about like the problems with vaccines that, you know, have bad side effects. But currently everything is about like, it did something, maybe not enough, so how do we kind of like push it a little bit further?
Thank you so much for the presentation. A question, with this approach, do you think is it possible to build kind of scalable solution? Because with personalization approach, basically you're kind of creating the vaccine for each individual, right? Mm-hmm. So you cannot have, you can aggregate data for millions of people, right? But it's not very accurate, a lot of false positives. So how can we build like scalable solution out of here? Or we need to kind of do it one-by-one basis, which is extremely expensive and slow, as we mentioned.
So I think that right now the scalability limits are mostly around the physical handling of stuff and the making of the vaccine. So we have like a lot of problems where, you know, a tumor sample kind of goes missing and we have to like email a lot of people to say like, where'd you put that piece of tissue? And that's not scalable. I mean, that's like, if you need 10 emails per patient to find the tumor sample, then that's not going to scale to 1 million people. The sort of technological bottleneck right now is manufacturing. And I think there are some ideas out there as far as making DNA vaccines instead of how we make them that could scale better, but it's still going to be pretty limited. I think that barring some, I think the sample handling and sequencing stuff can be really automated by a well-funded company. I think the making the vaccines really quickly is more of a waiting for research breakthroughs problem. That's it.